Dear all, welcome to our channel. In the previous lecture, we have discussed about the amino acid general structure and their properties. We have seen that all the amino acids are called as alpha amino acid because the amino group and the carboxyl group is attached to the same carbon. So that's why they are called alpha amino acids. And we also discussed that in the protein there are specifically the L isomers are present. So now in this lecture we will discuss about the classification of the amino acid. So the classification of the amino acids are majorly based on the R group. So R groups are further classified into two categories based on the solubility of these R group in the water. So they can be a non-polar and the non-polar R group. They are further classified based on that whether they are non-polar containing the aliphatic R group or the aromatic R group, or if they are polar, so they are charged, uncharged, or the positive charge group is present. So these R group vary into the structure and the size, and that makes the different type of the amino acids. Further, the amino acid can also be classified into the based on the metabolic fate, whether they can convert into the fatty acid or they can convert into the glucose. Based on this, these are also classified. They are called as ketogenic and the glucogenic amino acids, and amino acids are further classified into essential, non-essential, and the semi-essential amino acid based on the requirement of these amino acids from the food. So first we will discuss about the classification of the amino acid based on the R group. So as we have seen that non-polar amino acids, they are can be containing in the R group, that is the aliphatic side chain or the aromatic side chain. So here we are discussing about the non-polar amino acids that are having the aliphatic side chain. So R group in this class of the amino acids are non-polar because they are containing a long the hydrocarbon chain. So they are show the property of hydrophobicity. So these molecules, such type of the amino acids show the hydrophobicity and show tend to away from the water. So these type of the amino acids are the alanine, valine, leucine, isoleucine, methionine, and the side chain of these amino acids are then mostly you will find that the side chain, due to the side chain, they form a hydrophobic interaction. These amino acids are mostly you will find inside a protein where they are remains away from the water, and they due to this they stabilize the protein structure. They are hydrophobic in nature, so they are buried inside a protein structure and they are stabilize the protein through the hydrophobic interactions. Next, then if you will see that which among these there is a methionine amino acid, which is one of the two sulfur containing amino acid. Another sulfur containing amino acid is the cysteine. So the methionine and the cysteine are the two amino acids which are called as sulfur containing amino acid. So here in the methionine it is containing the thioether group. Next comes to the proline. Proline is also a aliphatic containing the aliphatic side chain, but it is having a distinct cyclic structure. It is another like it is a specific amino acid which is containing the amino, which is called as amino acid, because here the amino group is form a covalent bond with the R chain. Due to this unique side chain, it is the alpha amino group is attached to this side chain and form a 5-member ring structure. And due to this, the proline is less flexible, makes the proteins less flexible than the other amino acids. And this specific cyclic structure of the proline cause the folding of the protein, because that makes the less rigidity of the protein, and where the proline is present, there it is fold back. So this proline is having a very important role into the structural conformation of the protein. So let's see the structures of these amino acids. So these are the non-polar amino acids which are the which containing the aliphatic R group. So if you will see the glycine is not it is also considered in this category non-polar aliphatic R group, but it doesn't have a contribute so much for the this hydrophobicity because it is containing the hydrogen atom only. So, but other amino acid like alanine, it is containing this hydrocarbon chain, the methyl group, and this we are discussing about the proline. Proline here, it is called as the amino acid because their amino group is present here, and this there is, you can see this 5 members cyclic structure is formed for the proline, where this side chain is covalently linked to the amino group. Due to this, this is make a rigidity to the proline, and proline is mostly found into the band structure of the protein. And then this is valine, leucine, isoleucine, and the methionine. So these all amino acids, these are containing the aliphatic R group, that's hydrocarbon chains, that's contribute their hydrophobicity. So due to this hydrocarbon chains, these amino acids show these amino acids are considered as non-polar, and they are mostly found inside the protein, in the globular protein. They are mostly found into the inside, buried inside the protein, and where they cluster together and they stabilize the protein by the help of the hydrophobic interactions. Whereas this methionine, this is specific property, it is the one of the sulfur containing amino acid. Another sulfur containing amino acid is your cysteine, and this group is called a thioether group. Now the other amino acids, which are the non-polar aromatic amino acids, are relatively less non-polar compared to other aliphatic, but these are also participate into the hydrophobic interaction. So the amino acids which are considered as non-polar aromatic amino acids are phenylalanine, tyrosine, and the tryptophan. And this phenylalanine, here it is containing the phenyl group, it is containing the phenol group. So, and it is here is the tryptophan. Tryptophan is containing the indole group. So you can see that because the tyrosine and containing this hydroxyl group, so it can form a hydrogen bond, and it is found as a functional group in many enzymes. And tryptophan is also contribute towards some polarity, because due to this the nitrogen into this indole ring. So we can say that tyrosine and the tryptophan are significantly more polar in comparison to the phenylalanine due to this hydroxyl group and the nitrogen of the tryptophan indole ring. So these three are the aromatic amino acids, which are, we have to remember that phenylalanine, tryptophan, and the tyrosine. Sometimes questions are directly comes among, like if there are options are given, among which one is not a aromatic amino acid or which one is the aromatic amino acids. So phenylalanine, tyrosine, and the tryptophan, these are the three amino acids which contain the aromatic side chain. Now, this earlier we have discussed about the non-polar amino acids which are containing the hydrocarbon chain in the form of like aliphatic hydrocarbon chain or the aromatic hydrocarbon chain. Now we comes to the polar uncharged group. So R group of these amino acids are more soluble into the water. Why they are more soluble into the water? Because they are containing this hydroxyl group, which can form a hydrogen bond with the water. So the polarity of these two amino acids, which is serine, threonine, is contributed by the hydroxyl group present into their structure. Whereas in the cysteine is another polar R group. Here the polarity is contributed by the sulfahydryl group. So these amino acids can form a hydrogen bond with the water, whereas the polarity for the asparagine and the glutamine is contributed by the amide group. So these five amino acids are considered as the polar uncharged amino acids because they are having a polarity. They can form a hydrogen bond with the water, so they are solubilized. They can solubilize into the water. They are found mostly on the surface of the protein, so they can interact with the cytoplasm of water. And the name of these amino acids is serine, threonine, cysteine, asparagine, and the glutamine. So the very important here is the which group cause the polarity for these amino acids. So the hydroxyl group of the serine and the threonine cause the polarity of the amino acid, whereas the sulfahydryl group of the cysteine cause the polarity for the polarity of the cysteine amino acid. But the cysteine amino acid can form having a specific characteristic that it can form a covalent bond by the oxidation of the two cysteine residue. So, and that form a disulfide bridge where it can help into the very important role into the protein structure. Now, the most hydrophilic, the among the previous polar R group, there are some other amino R group which are either positive charge or the negative charge. And these positive charge or the negative charge amino acids are more polar compared to uncharged polar group because they are having some charge at pH, biological pH. So either they are having a positive charge or the negative charge. So these are the three amino acids which are having a positive charge R group at the biological pH, and the example is lysine, arginine and the histidine. So lysine is the another characteristic for the lysine is that it is having a secondary amino group. It is having a secondary amino group which is attached to this epsilon carbon, which is attached to the epsilon carbon. So this is the secondary amino group. So this question is that which amino acid is having the secondary amino group? So this is the lysine, and it is the positive charged amino acid. Another amino acid which is the arginine, which is containing the guanidino group, and it is also called, it is having a positive charge at the biological pH. And another is the histidine. Histidine is having a imidazole ring, and it is also the positive charge amino acid, and it is involved in many enzyme catalyzed reaction because the histidine residue facilitates the reaction by acting as a proton acceptor or the proton donor. In this way, it is helping to the many enzyme catalyzed reaction. So this histidine is involved in many enzyme catalyzed reaction. So these we have to remember this. These are the three amino acids. The lysine is containing the secondary amino group, and arginine is containing the guanidino group, and the histidine is containing the imidazole ring. And these all three amino acids are called as positive charge amino acid. Now comes to the two other amino acids, which is aspartate and glutamic acid. These amino acids, in contrast to the previous one that are having the positive charge group at the biological pH, these amino acids are having an extra carboxyl group. Due to this, these amino acids are having a negative charge at the biological pH. So due to the second carboxyl group, these amino acids are having a negative charge at the biological pH 7. So these are the two amino acids which are the negative charge. What are these? Glutamate and the aspartate. So these amino acids which are the, like we have discussed that these are the three amino acids which is phenylalanine, tryptophan and the tyrosine. These are called as the aromatic amino acids. There is a specific property of these amino acids. They absorb the UV light at 280 nanometer. So what are these two amino acids which specifically absorb the UV light at the 280 nanometer? That is tryptophan and the tyrosine. You can see the strong peak of the tryptophan and the tyrosine. You can see the very strong peak of the tryptophan and the tyrosine at the 280 nanometer of the UV light. So this property help the researcher to determine the concentration of the protein by utilizing this property. So if they, based on this, they see how much absorption is taking place by the protein. So based on this, some calculations, the concentration of the protein can be determined. So these are the two amino acids, that is the tryptophan and the tyrosine, which absorb a strong UV light at the 280 nanometer, and to a certain extent UV light is also absorbed by the phenylalanine. So and this help into the characterization and the concentration determination of the protein. So this property that we have to remember, that which amino acids absorb the UV light, that is the tryptophan, tyrosine and to a small extent the phenylalanine. Specifically it is I ask for the 280, then tryptophan and the tyrosine. The phenylalanine is show the bond around the 240 nanometer. Now comes to that we are discussing about the cysteine. Cysteine residue is, cysteine amino acid is having a specific group, that is the called as sulfahydryl group. So under the cellular environment, this cysteine can readily oxidize to form a disulfide bridge. You can see there is oxidation of the two there is oxidation of the two cysteine residue takes place, and there is a double bond. There is a cysteine bond is formed. Cysteine is formed, which is having a disulfide bond through which these two cysteine amino acids are linked. So this structure help into the structure formation of many multimeric proteins. Like you can see the structure of the immunoglobulin, where you will find this is the light chain and the heavy chain of the of the immunoglobulin, and these light chain and the heavy chains are linked with the help of this disulfide bond. This disulfide bond you will find here, and due to this, these polypeptide chains are held together. So this cysteine residue is having a very important role in the protein structure. So this cysteine is also found in the insulin structure help. This is, you will find the cysteine in the here, where the creatine protein, this cysteine residue is oxidized to form a disulfide bond. So now we have discussed what are the amino acids and how they are classified based on the R group, and specifically we have discussed based on the polarity. So we have seen that their amino acid can be classified into polar or the non-polar. If they are non-polar, then whether they are having a aliphatic side chain or the aromatic side chain. So what are these aliphatic side chain? Specifically, there are the alanine, valine, leucine, isoleucine, methionine, and the proline. And glycine is also comes under the same category, but it doesn't, because it is having a hydrogen atom, so it doesn't contribute too much towards the hydrophobicity. And then comes to the non-polar aromatic amino acid. These are the phenylalanine, tryptophan, and the tyrosine. Then we have discussed about the some amino acid which are having the polar uncharged group. That is the serine, threonine, then asparagine, and the glutamine and cysteine. Then we have discussed about the some amino acids which are the polar but positive charge. So that is lysine, arginine, and the histidine. And then we have discussed about the polar negative charge, that is aspartic acid and the glutamic acid. So this is the categorization or the classification of the amino acid based on the R group, and specifically on the polarity of the R group. And you can see there is some abbreviation is also used for these amino acids. Like glycine is, you can see there are 3-letter abbreviations are used for this amino acid instead of the complete name. Glycine is termed, the abbreviation is GLY, glycine. Alanine is ALA, and then there is also a symbol is also used. So that glycine is abbreviation is GLY and symbol its use is G. For alanine simply that is ALA and the symbol is A. Proline PRO and the symbol is P. Valine VAL, so symbol is V. Leucine LEU, symbol is L. Isoleucine abbreviation is ILE, symbol is R. Methionine abbreviation is MET, MET, and the symbol is M. Aromatic amino acid is phenylalanine, FHE, and its symbol is F. Tyrosine TYR, symbol is Y. Tryptophan TRP, and the symbol is W. Serine SER, symbol is S. Threonine THR, symbol is T. Cysteine CYS, symbol is C. Asparagine. So you can see that there is some you can directly, first letter is not used in some cases, that is asparagine, where it is N. And the glutamine, that is the first letter is not used here because it is already used for the glycine, so here it is Q. Tryptophan also, that is W. For the lysine, that is K. Histidine, it is for the H, and the arginine, it is R. And the aspartic acid ASP, glutamic acid, and that is D and E. So these are the abbreviations and the symbols which are used when the amino acid are shown in a peptide sequence. So it is a simple way to show these amino acids in an alphabet. So now comes to the some. These are the earlier amino acids which we have discussed. These 20 amino acids are called as the common amino acid or the standard amino acid. Now comes to the some amino acids which are called as uncommon amino acids. So these are the here after these amino acids you will find that these are amino acids which are more created after once they has the amino acids in is amino acid from the standard amino acid is incorporated into a protein, then there is some chemical modification takes place and these amino acids are synthesized. And apart from these two other amino acids are also found here newly identified amino acid, that is the selenosystine and the pyrrolysines, which are incorporated by the into the proteins in a normal way, but by the using the stop codons that we will discuss in the coming lecture. And the other amino acid you will find that these amino acids are present in the living organism, but they are not the constituent of the protein. Like they are not a work. They are, the proteins are not made up of these amino acids, but they are the containing this amino and the carboxyl group, so they are considered as the amino acids. So now comes to the amino acids, which are in like we have discussed that there are certain amino acids which are created by the modifications of the common residues. Like a protein is synthesized and the common amino acids like proline or the lysines, they has been incorporated into a protein. Then after incorporation of the protein, there is some chemical modifications takes place of these amino acids. So like example you can say is the proline, where the hydroxyl group is incorporated, that is added into this proline. So it is the 4-hydroxyproline. So this is a uncommon type of the amino acid, and it is by the chemical modification once the common amino acid has been incorporated into the protein. And the 4-hydroxyproline that is found into the plant cell wall, where these proteins which this amino acid you can find that is in the plant cell wall. Next is the 5-hydroxyl lysine. So 5-hydroxylysine and the 4-hydroxyproline, these both amino acid you will find into the collagen protein, which is the component of the connective tissue. Another so here you can see hydroxyl group is added to the 5th carbon. So it is the 5-hydroxylysine. And the 6-methyl lysine, there you can see the methyl group is added. Methyl group is added to this amino group. So it is the 6-N-methyl lysine. So this methyl lysine, that we have to remember this thing. Like questions are comes, this 6-methyl lysine you can find in which type of the protein. So it is in the myosin protein, where the 4-hydroxyproline and the 5-hydroxylysine, that you can both type of the protein, both type of the amino acids are you can find in the collagen protein, whereas 4-hydroxyproline is found into the in the plant cell wall. So there, since these are protein, some protein you will find these type of the amino acids. Now another amino acid which is created by the general modifications of the protein, that is the gamma carboxyglutamate. Gamma carboxyglutamate is a amino acid which is incorporated by the carboxyl group into the glutamate. So in this you can find that in this protein, this amino acid you will find into the blood clotting protein prothrombin. This protein is involved into the blood clotting formation and some other proteins, where the calcium is a part of the biological function. So this gamma carboxyglutamate you can find the gamma carboxyglutamate. So this carbon is alpha, this is beta, and this is gamma. So gamma carboxyl glutamate. So this type of the gamma carboxyl glutamate, you will find that this amino acid is the component of blood clotting protein prothrombin. Another protein amino acid that is found is the in the elastin. In a that there is specific amino acid which is called as a desmosine, and it is this desmosine is formed by the combination of the 4 lysine amino acid. So you can find the desmosine is found into the elastin protein. This protein is found in a connective tissue such as skin and the lungs, and its cross-linking in the elastin. That desmosine provide the elasticity and the tensile strength to this protein. So this is a specific type of the structure is for the desmosine that you will find is specifically into the elastin protein. So these amino acid that we have to remember, these is placed with this chemically modified amino acid in found in which type of the protein. And these amino acids are find like these amino acids are created by the chemical modifications. Once these like glutamate is incorporated into the protein, then after the incorporation of the glutamate, the carboxyl group is added to the alpha to the gamma group, gamma carbon, and then there is a formation of gamma carboxy glutamate. And this gamma carboxy glutamate is specifically bound to the protein prothrombin. Now comes to that we have discussed the 20 amino acids which are the common proteogenic amino acid we also called as. So apart from these 20, there are other 2 amino acids which are recently identified. These are also introduced during the protein synthesis. Earlier we have seen that some amino acids which are created through the post synthesis modifications, but these amino acid is introduced during the protein synthesis. So these we can say these are also the we can say the 21 amino acid. But and so you can see that selenocysteine and the cysteine. If you will see the structure of the selenocysteine, this is the structure of cysteine. Selenocysteine is containing a selenium group. This is containing the selenium group instead of sulfur. So this is the difference between the selenocysteine and the cysteine. Otherwise, they are both are very similar in their structure. Selenocysteine is derived from serine. It is not derived from cysteine, and it is also a component of very few protein. So the selenocysteine is considered as a 21st amino acid because it can be 23rd amino acid which is proteogenic in nature. So it can be incorporated into the protein, and it is found that both eukaryotic and the prokaryotic system can be incorporate the selenocysteine into the protein, but it required a specific sequence that is known as ACCIS sequence. So this ACCIS sequence is known as the selenocysteine insertion sequence. And this ACCIS element is specifically found into the RNA which direct this the UGA codon to use as a selenocysteine codon. Otherwise, the UGA codon is a stop codon. So if you will see the structure also, the serine of the selenocysteine, then it is very similar, and here only there is a change of oxygen atom with the selenium atom. So it is very important to know that selenocysteine is derived from serine, and the element which is required to incorporate that is HCCIS, and which codon is used here, that is the UGA codon. So these few questions are like very important from this selenocysteine topic. Now comes to the pyrrolysine. The pyrrolysine is also a modified lysine, and it is also considered as a 22 proteogenic amino acid. And in it is how it is created. It is created by the modification of the lysine, where a 4-methyl pyrroline 5-carboxylate group is attached to the epsilon amino group. As we know that lysine is having a additional amino group which is present at the epsilon position. So to this group is attached, and it is encoded by a UAG. UAG here is again a stop codon. And this pyrrolysine was discovered in 2002 from some methanogenic bacteria. That bacteria that produce the methanogen, that the example is Methanosarcina barkeri, and it is found at the active site of methyltransferase enzyme. First, it was identified from this enzyme active site, and it is used in the biosynthesis of some methanogenic bacterial protein. And it is found that in the human this pyrrolysine is not present. So these we have discussed about the amino acids. So we can say there are 22 amino acids which lead to the synthesis of a protein, which are directly incorporated into the protein, and some amino acids which are like modified by some chemical modifications. And there are some other additional 300 additional amino acids which are found into the cells, but they are not involved into the protein constituent as a protein constituent, and but they are having a different functions like ornithine and the citrulline. These are the example is where these are the two amino acids which involve into the biosynthesis of arginine in the, they are involved in the urea cycle. So these are work as a key metabolite. Similar in this way, there are many additional amino acids are found into the cells, which are having a different type of the function, but these are not a constituent of amino of the protein. But these amino acids, because they are considered as amino acid, because they are having a amino in the carboxyl group. So this classification which we have discussed is based on the like first on the polarity of the R group. Now if you will come and then the amino acid can also classified based on the some metabolic fate. So these amino acids which we have discussed, the 20 amino acids, they can be classified as a ketogenic amino acid or the glucogenic amino acid. So what is the ketogenic amino acid? So ketogenic amino acids are those amino acids which produced acetyl coenzyme A or the acetoacetyl coenzyme A. So they can lead to the production of the fatty acid or the ketone bodies. So such type of the amino acids where they can lead to the production of acetyl coenzyme A, where it can be converted into the fatty acid, or it can lead to the production of ketone bodies. Such type of the amino acids are called as ketogenic amino acid, and these amino acids cannot be converted into the glucose. So this example of the ketogenic amino acid is leucine and the lysine. These are the two amino acids which are specifically called as ketogenic amino acid. They cannot be converted into the glucose, whereas some amino acids are called as glucogenic amino acids, where they produce a pyruvate or either the TCA cycle intermediate, such as alpha ketoglutarate, succinyl coenzyme A, fumarate, or the oxaloacetate. These are all are the TCA cycle intermediate, where it is again converted into pyruvate, and through the process of gluconeogenesis, it can lead to the production of the glucose. So such amino acid where their skeleton can be used for the production of pyruvate or the TCA cycle and ultimately can lead to the production of glucose are called as glucogenic amino acid. And these are the example of the amino acid which are considered as the glucogenic amino acids. That is glycine, serine, aspartic acid, glutamic acid, glutamine, valine, methionine, histidine, and the arginine. Whereas some amino acids which can produce both ketogenic and the glucogenic amino acids are also present. So these are the amino acid example is isoleucine, tryptophan, phenylalanine, tyrosine, and the threonine. So these amino acids can produce a compound which can be lead to the production of either acetyl coenzyme A, or they can be converted back into the glucose. Now comes to the classification of the amino acids based on the nutrition. Like these amino acids can be classified into a three category, that is essential, non-essential, and semi-essential, or we can say conditionally essential amino acids. So essential amino acids are those amino acids which our body cannot synthesize, and we have to acquire these amino acids through our food, because every amino acid is required for the protein synthesis, and so thus if there is a deficiency, the body cannot make these, so there will be deficiency of these amino acids. So 9 of the 20 amino acids cannot be synthesized in our body, and we call them as essential amino acid. We have to take them in the form of a food from the food sources. And the other amino acids which are that 11 amino acids are called as non-essential amino acid, because our body can produce these amino acids. So if you don't take them from food, there is a supply of these amino acids through by our body. So these amino acids, the example of these amino acids is these alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. And there are some amino acids which are further called as semi-essential or the conditionally essential amino acids, because although some non-essential amino acids, they are called as semi-essential or conditionally essential amino acids. That example is arginine, cysteine, and the tyrosine, because their concentration becomes low during a certain conditions, such as during the infant, or we have either we are get sick or injured. In these conditions, their concentration of these amino acids, that arginine, cysteine and the tyrosine, which are otherwise the non-essential amino acids, but we have to supply, their concentration becomes very low, so we have to supply from the in the form of food. So these are considered as the semi-essential or conditionally essential amino acid. So now we have discussed the classification of the amino acids based on, first we have discussed that the amino acids which are the 20 amino acid which has been classified based on polarity. They are further classified in the polarity, that is polar and non-polar. Polar aromatic amino acid, aliphatic amino acids. Then polar charged, polar uncharged, and the polar charged. Then again two types: polar positive charge and the negative charge. Then we have seen there is, apart from the 20 amino acids, these are some non-standard proteogenic amino acids are also found, which are called as 21 and the 22 amino acids, that is selenosystine and the pyrrolysine, which use the stop codon as a genetic code, and these are not a component, general component of the protein. These are found that they are specifically found in very few protein, component of very few protein. Then we have seen that some amino acids which are chemically modified. Once the like normal amino acids has been modified, then these amino acid residues are chemically modified too, and then these are component of some protein. The example we can take, that is the gamma carboxyglutamate. Then we have seen that how these amino acids can be further classified. These amino acids can be further classified based on the nutrition and the metabolic fate. Like metabolic fate, that is ketogenic amino acid, which can be like the skeleton system can be converted into fatty acid, or the fatty acid by the conversion of acetyl coenzyme or acetoacetyl coenzyme, where some amino acids, that is the glucogenic amino acid, where they can be converted into pyruvate or the TCA cycle intermediate. Then some amino acids are the amino acids are further classified based on the nutrition source, like essential, non-essential and the semi-essential amino acids. So this is about the classification of the amino acids. Next class we will discuss about the peptide bond formation and the different structure level of the protein. Thank you for watching this video. If you like the video, please like and subscribe the our channel. Thank you. We'll meet into the next lecture.